Why are cells in our body different if they all have the same DNA? When that sperm fertilizes mom's egg, they fuse together and form the first cell from which our entire body is formed. This cell is called zygote and contains a mixture of DNA from both parents, 50% of mom's DNA and 50% of dad's DNA. The zygote is a stem cell, which, like the stem of a plant from which the rest of it branches, has the ability to originate all the tissues and organs of the body, and also the placenta. The zygote goes through segmentation and multiplies, forming the blastomeres, and after a few divisions, it forms the morula, and finally the blastocyst, a mass of cells that contains an internal liquid cavity which helps to distinguish two cellular populations. The trophoblast, or external cellular layer, which encloses the blastocyst and ends up forming the placenta, and the embryoblast, or inner cellular mass, which becomes the embryo. The cells of the embryoblast are denominated embryonic stem cells, or ESCs. They are considered pluripotent because they can give rise to all the cell types in a fetus, as they still got almost their entire DNA unlocked. The IPSCs, generated from somatic cells, emulate ESCs, unlocking their DNA so pluripotentiality can be achieved again. But what do I mean by unlocked? Our DNA is like an instruction list with all the molecules needed in the body to operate, but not all the molecules are needed in each one of the organ tissues. Unlike protozoa, whom are unicellular eukaryotic cells which use all their DNA because they are unicellular and they have to fit all their body parts and weapons in only one cell, and by weapons I mean paralyzing darts and crazy stuff like that, so they can pierce, neutralize and eat their prey. Humans and all the multicellular organisms need their cells to become specialized so they can save energy. The pancreas makes insulin, so their cells have to keep the insulin gene or book with insulin manufacturing instructions open. In contrast, the rest of the cells in the body do not need to express insulin. So, through a process known as epigenetic changes, they lock the genes they don't need. Epigenetic changes get maintained through cell divisions, and they are responsible for the cells to be specific in the functions they have to do. Maybe you're guessing it. IPSCs are obtained by removing these epigenetic marks, unlocking cells' potential to become everything they've dreamed to be. On this channel, I'll share with you the news about regenerative medicine, genome editing, and tissue engineering. Subscribe to keep in touch, and remember that the fact that it seems impossible is what makes it so beautiful.